to start recording. Okay, now it has recorded. Fine, no problem. So those who have joined just now, if you guys want me to go back to few slides, you guys can tell me. Okay, I can just quickly revise those slides. So as I was saying, that the most important design criteria for a TEMP arise from observations about normal biological function of the tissue to be replaced. So before designing any tissue engineered medical product, like for pancreas or for skin or for liver, you first have to understand how that organ or how that tissue system functions on its own, like in a normal setting. So and also you have to take care of few fundamental facts, like what is the cell number that you need, okay, in your product. Then cell function, what cell function you want to mimic in your product. Then cell to ECM distribution. So for cartilage we have seen that it is the cell density of the chondrocytes in cartilage is very less, but when you talk about skin, it is not so. Like the order of cell number is extremely different. So you have to know how many cells you need and cell to ECM distribution. So in cartilage mostly ECM is there, but in skin ECM percentage, cell to ECM ratio will be high, and in cartilage it will be low. So this kind of thing you should know prior to designing or any kind of tissue engineered medical product. Then you have to also know what is the mechanical property or the mechanical strength of the tissue that you're looking for. So for bone, the strength is very high, right? Young's modulus is very high. But when you talk about soft tissues like liver or heart, the strength that you're going to look for is going to be less. And adaptability of tissue to biological stimuli means how your tissue engineered medical product is responding to some kind of biological stimuli, like electrical stimuli. So people have made artificial heart tissue, where the cardiomyocytes, they are responding to electrical impulses and kind of showing that beating effect, pulsating effect it is showing. So these kind of stimuli, how they are responding, you have to think and you have to know from the normal organ function. So for example, a comparative analysis of in vivo beta islet insulin production to insulin production in vitro would be necessary. Okay, so if we are looking for going to design beta islet cell encapsulation kind of a product, so we have to first see in encapsulated mode how much insulin is being produced in vitro, okay, and whether it is comparable to the insulin production that takes place in vivo. Additionally, you need to complete the examination of all the methods that is used to increase insulin production capability. Means you have to search for literature and you have to see what all methods people have applied to increase insulin production in vitro, and also test how the primary islet cells or beta islet cells are responding to the various glucose level, okay, in vitro. All the testing in tissue engineering happens in vitro, okay, and then people go in vivo, and then only you will have some concrete data that you will show when you are going for approval, regulatory approval for this kind of a product, and then it will come to clinical setting. So this kind of thinking or the mindset you need to develop while designing a tissue engineered medical product. So, as I was saying, that you should know about the cell numbers, right? So we'll talk about that now. Cell density in human tissues are on the order of 1 to 3 billion cells per million per ml. Okay, so in human tissue, like this is a general estimation of cell number. It will definitely vary from tissue to tissue, but in general, in a human tissue, there is about 1 to 3 billion cells per ml. So if, for example, the volume of a 70 kg human is about 70,000 ml. So you just have to multiply it with the density of water. So from that you will get the volume, that is 70,000 ml. So in that volume, then the human body will consist of around 100 trillion cells. So here this calculation is basically they have multiplied the volume of the human body with the cell density that is present in a human tissue. So in this way they have calculated that in a human body, complete human body has approximately 100 trillion cells. Okay, that is 10 raised to power 12 is trillion. Now a typical organ is like approximately is of 100 to 500 ml in volume. So what will be the cell number in an organ? So you will just have to multiply it with the cell density in a tissue, and you will get that there is around 100 to 1500 billion cells in an organ only. So now organ is composed of various functional subunits. Like kidneys are made up of nephrons, and nephrons are called functional subunits. So each organ has a functional subunit, and their linear dimension, they are saying, is approximately 100 micron. So if you make a cube, okay, cube with each side of 100 micron, and you get the volume of that cube. So it will be you can get the volume, and for that volume you can get how many cells will be there, okay, when you multiply it with the cell density. So they are saying that in a functional subunit there will be approximately 500 to 1000 cells. So how many functional subunit will be there in an organ? So they have just multiplied with the number of cells, like 5000 to 500 to 1000. They have so divided it with this number, that is number of cells. So they are saying that in a typical organ there is around 100 million functional subunits, or 10 raised to power 8 functional subunits. So these are the typical numbers. Okay. Okay. Am I audible? I think momentarily I left the meeting. Am I audible now?
Yes, ma'am.
Yeah, okay, thanks. So as I was saying, so for making TEM, one needs to opt for a tissue culture technique which will allow production of around million to billion cells. So 10 to the power 6 to 10 to the power 9 cells you need for making a tissue engineered medical product, because you are mimicking the cell numbers that are typically present in a human tissue. Okay, and if you are going for the organs, so you have to mimic those cell numbers. So what cell culture technique that you are using is going to play an important, very important role. And this million to billion cells is actually called clinically meaningful cell number. Okay, anything less than that you are just doing in vitro thing, but if you want to mimic the human tissue you have to go for these cell numbers. So in this table, as you can see very nicely, um, these things are summarized. Okay, so whole body consists of 10 to the power 14, that is I said 100 trillion cells. If you do the math you will get 10 to the power 14 cells in a whole body. Okay, and a human organ consists of 10 to the power 9 to 10 to the power 11 cells. And whereas a functional subunit, which an organ is composed of functional subunit, and so each functional subunit consists of 100 to 1000 cells. So these are the cell numbers that you see in people. So if you want to develop a therapy, okay, so you need to mimic those numbers. So this is called clinically meaningful cell numbers, and that is 10 to the power 7 to 10 to the power 9, or 10 to the power 6 to 10 to the power 9 million to billion cells you need. Okay, and the tissue culture technique that you are using for this production should be optimized to generate that many kind of cells. Now, what are the fundamental limitations to the production of primary cells? So, so still now you are like, you must have understood. So for making a tissue engineered medical product, the main important component is the cell. Okay, so not all cells in our body can be cultured in an equal manner. Means all cells do not possess the same proliferation rate. So there are some limitations. So we'll talk about those. The number of cell divisions that a primary cell undergoes is subject to the so-called Hayflick limit. So Hayflick limit is basically the limit of it provides a limit, means to how many cell divisions a primary cell can undergo. So number of cell division is totally different from the number of passages. Okay, don't confuse in between these two things. Uh, okay, if you are not still clear about these two, you can ask me, but I'm assuming that you're able to differentiate between the cell division and number of passages. Okay, the primary human cells can undergo about 30 to 50 doublings in culture. Doublings means one cell divided into two. Okay, so one cell can like double 50 to 30 to 50 times. Okay, so if I ask you how many cells you will get after 50 doublings, how will you calculate? Anyone? Is anyone there? Hello
Yes,
guys,
ma'am.
am I is my question clear to you? My question is how many cells you will get after 50 doublings? How will you calculate that? Anyone would like to try?
Ma'am, the result will be exponentially 50 times more as usual.
Right. It will be a like something raised to the power something, right? Power रहेगा, exponent रहेगा. So can you tell me that figure? What will be if I ask you the number, just number, how many cell number you will get? You don't have to calculate on your calculator. Just tell me some like what will be the base and उसका power कितना रहेगा?
It can be like 50 into 10 power 10 or 10 power 15, because a single cell can produce that much.
Okay. How did you come to that calculation? Can you explain?
But it has, as per the screen, it mentioned a single cell can produce that much.
Hmm.
So after 50 doublings, a single cell's uh,
How many
product
cells you
can
will
be
get?
that much. How
So
many cells?
after 50 doublings, how many cells you will get from a single cell?
Ma'am,
So
10
sir,
raised to the power 10 raised 10 raised to the power 50, 60 to 10 raised to the power 65.
Okay. So why did you take the base 10?
Ma'am, because they are common. The exponential power is just increasing, so just why I added.
Okay. So a single cell can produce these many cells, right? So here it is 10 square 10 to 10 square 15. You're seeing in the screen. So how did they come to this value? I'm asking that. They must have multiplied something, right? Or you know. So let me just not be. I think you guys are confused. So it's very simple actually. One cell can divide and produce how many cells? Two cells. Am I correct? Right? Am I
Yes,
correct?
ma'am.
Yeah. So it's a geometric thing, means then
Pressure.
again one daughter cell will divide into two, then another daughter cell will divide into two. So two should be the base, correct? And then how many doublings are going to happen? If there are 30 doublings, it will be 2 raised to power 30. If there are 50 doublings, 2 raised to power 50. Now if you put that value in a calculator, then you will get 10 raised to power 10, 10 raised to power 50. Like approximately you will get that. Am I clear?
yes, ma'am.
Yes.
Yeah, it's easy, right? So from there you are going to get this value 10 to the power 10 to 10 to the power 15 cells. If you if you get like 2 raised to the power 30 or 2 raised to the power 50, is it all everyone understood this calculation, right? Okay, fine. So as you can see, this number 10 to the power 15 10 to the power 10 means this number of cells, it is more than sufficient for making a tissue engineered medical product. Because what I told that we need 10 to the power 6 to 10 to the power 9 cells, like million to billion cells, for making a product. And a primary cell can give more than enough cells. So the Hayflick limit is not a limitation here. But again, I would like to repeat that different cells in our body have different proliferation potential. Okay, so you have to take care of that. So dermal fibroblast means the fibroblast present in the dermis layer of skin. Those cells, as well as the skeletal myocytes, they grow very well. Okay, they grow very well in culture, so they can easily produce 10 to the power 10, 10 to the power 15 cells. But there are few cell types like hepatocytes, then beta islet cells of pancreas. They grow very limitedly, or they do not grow at all. So if you are going to make a product, liver based product, or pancreas, a product for the pancreas ailment or anything like that, you need to think how you will get those many cells, like million to billion cells. So there comes the problem. Also, primary cell cultures, they often contain various cell types. So you know, right, in any tissue there will be one major type of cell type, like the fibroblast or keratinocytes, as I told, in the skin. But there are also other cell types like supporting cell types. Means there can be blood cells, there can be the immune cells like macrophages, right? There will be few nerve endings. So if the lymphatic vessels are running, okay, so there will be lymphatic cells as well. So it is, one tissue is not composed of one or two cells, there are multiple cells present. So when you are going to make a primary culture directly from the patient, it's going to be a very challenging task to get only one cell population out of say 5 6. Okay, and you may say like I will use the surface marker, but it is also possible surface marker that you are choosing, it is expressed by one or two cells. So you have to be very thorough in your isolation procedure. So obtaining pure cell culture is a challenge, as I implied. Okay. Also, primary cell cultures often undergo cell differentiation, and at the same time cell number increases, like for stem cells. So stem cells, like hematopoietic stem cells, if you do like in vitro cell culture, they will very easily differentiate. Okay, so first they will proliferate and then they'll differentiate. So if your goal is to use the stem cells only, you have to be very careful that they do not differentiate. But if you want them to differentiate, then it's not a problem. Okay, so these are the limitations that you have when you are going to produce around million to billion primary cells for your product, tissue engineered medical product. So this is the graph that shows the Hayflick limit that I told. That is the limit on how many cell division a primary cell can undergo. So on the y-axis is given the cell doublings. Okay, 50, 30, just we talked about. And here is the given, and the x-axis is given the age of the donor. Means from the person who which the cell is taken. Okay, the primary cell is taken. So if you take the cell from a fetus or from a very young baby, so since those cells are young, they can undergo like approximately 60 to 70 doublings. But if you take the cell from a old man, like from who is in the age of 80 to 90 years old, their cells will have the potential to only go up to 40 to 50 doublings. So this kind of limitation exist, okay, and the number of cell division is influenced by the age of the donor, okay. And this is the line, if you do a linear fitting, from this linear fitting we can say on an average or on approximately a primary cell can undergo 30 to 50 doublings, and this is the Hayflick limit, okay. We will talk about this, like why this limitation takes place is actually molecular level of phenomena happens, because of which this cell doublings decreases with increase in age, okay. We'll talk about those in that in subsequent discussion. So as I told you that primary cells vary greatly in their growth rates in culture. So hematopoietic progenitor cells, so these are the means when we'll talk about stem cells, there we'll talk about progenitor cells as well. So these cells, they can undergo around like one doubling time. What is the doubling time? Means one cell dividing into two, and that happens in around 11 to 12 hours. So which is the minimum time taken, or you can say these cells are undergoing very fast. I have a very fast rate of doubling. Whereas dermal fibroblasts, they have around 15 hours of doubling. But if you look at adult chondrocyte cells that are taken from their cartilage tissue, they grow slowly, rather slowly, or relatively slowly, with a doubling time of 24 to 48 hours. Whereas the hepatocyte cells taken from an adult human, they barely grow, okay, they barely grow in culture. And if you look at the cardiomyocytes, the heart muscle cells, they cannot grow in culture in any meaningful time. So our current ability to grow human cells in culture varies significantly with each cell type, okay. So you will see in literature that for hepatocytes or cardiomyocytes, they are using animal cells instead, like mice cell or like pig cell or something like that, bovine cells, to prove their concept, proof of concept, to show a proof of concept of their research. And also, but if you look at the skin cells, it is used for humans only. They show all the things from human cells only. Cells are mostly produced by using standard cell culture techniques, which uses either tea flask or bags. Okay, and you can use special cell culture chambers called bioreactor if you want to produce large quantity of cells. So bioreactors size can be varied. Okay, lab scale bioreactor comes, an industry scale comes. So there you can produce trillions of cells at a time. But if you go for lab scale, you can hardly go up to million, okay. Like if you grow one T75 plus, you will get around 3 to like 2 to 3 million cells. So, and once adequate number of cells have been produced, you can either directly use those cells for your product, or you can cryopreserve it, means preserve it in liquid nitrogen so that they last long. The cells must be delivered in a clinically meaningful numbers that we talked about to provide a benefit to the recipient. There is some clinical benefit that recipient should observe, okay, from this type of therapy. For example, the isolated hepatocytes, when injected into the portal vein for treatment of metabolic genetic defects, they engraft at a rate of approximately 10%. Okay, so if you have given around 100 cells, okay, and only 10% of it, okay, will engraft. Rest 90% they'll flush out, or they will be detected by the immune cells, macrophages and all, and it destroyed by phagocytosis. Okay, so in liver there the macrophages called Kupffer cells, so they will destroy it. So in order to increase this rate of engraftment, people use various kind of delivery systems like encapsulation, like we saw for beta islet cells. People have encapsulated beta islet cells for producing insulin inside the patient's body. Or you can use biomaterial scaffolds. So we'll talk about what are biomaterials and what are scaffolds. So for now, you just understand that scaffolds are artificial, support, okay, on which you are growing the cells, okay, or within which you are growing the cells. So you are kind of protecting the cells from the immediate contact with the uh, uh, from the immediate contact of the blood cells or the blood, okay, and it can reduce your chances of immune rejection. So these kind of strategies have been used in order to deliver adequate number of cells. So till now, does any one of you have any queries? Any queries so far?
Excuse me, ma'am.
Yeah,
Ma'am
yeah.
can
Who
like
is speaking?
encapsulation and other.
Who is
Ma'am
speaking?
Harshita.
Harshita. Okay, okay. Go ahead. Yeah,
Ma'am Harshita.
yeah, yeah. Go ahead.
Ma'am, like if we do stem cell therapy in a person suffering from autoimmune disease, ma'am will like encapsulation and masking out. Ma'am will encapsulation or any other thing will genuinely help or not.
So you're talking about delivering stem cells, okay, into a patient, right, for autoimmune disorders. So it will definitely help,
Yes, ma'am.
okay. It will, as I said, it will increase, chances of um, target. It will increase chances of targeted delivery. If you have heard this term, targeted delivery, like for drug delivery, we do targeted delivery. Similarly, if you want those cells to smoothly reach their target site, okay, without getting phagocytosed by the macrophages, okay, or getting recognized by the antibodies and all, so it will definitely help. It will definitely help.
Okay, ma'am.
Ma'am, I have a question.
Yeah.
Ma'am, you said we can't culture cardiomyocytes, but I have read somewhere that in vitro people have done beating cardiomyocytes.
Yeah.
Is that ever done, ma'am?
So yes, actually people have made artificial hearts. Actually, they have 3D bioprinted artificial hearts. So if you look at those kind of papers, they haven't used primary adult cardiomyocytes. What means they are either made into a cell line. If you know what I means they are transfected by a virus. So they are made cancerous and made immortal.
Okay, ma'am. Okay, ma'am.
So
Okay.
I'm talking about primary, taken directly from the body and then cultured into clinically
Okay,
used.
ma'am. Okay, ma'am.
So
Got it,
function,
ma'am.
so if you modify the cell line, function, what function that you want to see, some function will retain. Okay.
Okay, ma'am.
Any other queries so far? Okay. So, are there any queries from the last week's session? If you have, if any one of you have gone through the lecture slides, no. So, because I asked this because we are going to start with the next topic, okay, today only. So we have time, so we can do it. So the next topic is going to be rather simple and more into cell biology, okay, tissue and cell biology. So in our body, there are 10 organ systems. Okay, 10. So you can see from this table, five of them are listed here, like the circulatory, respiratory, digestive, okay, circulatory, respiratory, digestive, urinary, and musculoskeletal. So these are the 10 organ systems that are present in our body. Sorry, five, and rest five is theirs: immune system, nervous system, endocrine, reproductive, and integumentary system. So these are the 10 organ systems in our body, and each organ system is composed of on different kinds of cells, cell types, then tissue type, okay, and then blood vessels are connected. So we'll see here this hierarchy, how an organ system is made. So if we look at the very initial stage, when fertilization happens, or fertilization of ovum happens, so it will, as you all know, the gastrulation process happens, a fetus develop. So if you look at the cellular level, what is happening to the cells? They are rapidly dividing. Okay, they are going through cell doublings, and as they are doing that, they are organizing themselves to form various kind of differentiated cells, like you get epithelial cells, connective cells, connective tissue cells, nerve cells, muscle cells, okay, blood cells. This kind of differentiation happens, and once the cells get differentiated, means getting assigned to a particular function, they organize themselves to form a tissue. Okay, so as you guys know, in a tissue there are around billions of cells. So these are the different kind of tissue types that you can see. There's epithelium tissue, connective tissue, nervous tissue, muscle tissue. And when these tissue types they combine, they form the functional unit, functional subunit of an organ. And around 100 to 1000 functional subunits, when they come together, they form an organ. Like here a nephron, when 500 or thousands of these functional subunits come together, they form an organ, which is a very small organ, around 500 to 1000 ml of volume. And this organ is connected to an organ system where there are multiple organs. So there is urinary bladder, the kidney is connected to all the tubes and the ducts and all. So it forms an organ system. And when various kind of these kind of organ systems, as I told, there are 10 organ systems. These organ systems make a human, and who is totally functional and normal and healthy. So this is how the hierarchical organization of our body. Okay. So now you can understand why the cell numbers, knowing the cell numbers is important, so that you can imagine the scale of the magnitude of the things that is going on inside our body. And if in future, if you want to design a tissue, you have to take those things in account. So tissue basically consists of one tissue, any kind of tissue consists of two things, two important things. One is the cell type. Okay, what is the cells? How many different types of cells are there? Second is the extracellular matrix or ECM. So these two things are the major component of any kind of tissue. The cells, they are continuously producing and degrading the ECM, okay, so that they are organizing their environment, also called the microenvironment, according to their needs, okay. and uh, and uh, and overall, this kind of changes, these dynamic changes is giving the tissue function. The cells are producing the ECM, also they are degrading the ECM, and they are designing their own environment, okay. And when you look at the tissue overall from a like a very broader perspective, when you look at the tissue, it will gain some kind of a function because of the activity of each individual cells. So let's talk about ECM first. Like what ECM is composed of, what is the function of ECM, etc. So substantial part of the volume of tissues is made up of this ECM. So ECM is basically an interconnected network of macromolecules, okay, and it forms an ECM. So ECM, if you want to visualize how it looks like a jelly kind of a, it has a jelly kind of a texture, okay, like gelatin, something like that, within which the cells are basically floating and they are migrating and they are communicating and all. And this ECM can be composed of various kind of proteins as well as polysaccharides. So what examples, if I may say? So there is collagen, laminin, fibronectin, elastin, proteoglycans, hyaluronan, fibrinogen, glycosaminoglycans, or many other examples are there of the various kind of proteins and polysaccharides that make up the ECM. Also, you will see that there is types of collagen, there are types of fibronectin like 1, 2, 3, 4. Okay, and percentages vary, how much collagen should be there, how much fibronectin should be there. So and this composition of ECM is unique to each type of tissue. So a skin tissue will have a different composition, whereas a liver tissue will have a different ECM composition. All right. So and it varies. Why it varies? Because of what kind of cells are there. Okay, so as I told, cells are the ones who are synthesizing these proteins at the polysaccharides of ECM. They are the ones who are modulating how the collagen fibers should be oriented. Okay, or also they monitor the degradation of these collagen fibers, like by secreting various kind of enzymes. So they are completely managing their composition of the ECM. In turn, what ECM would do? ECM can exert control over many cellular fate processes, okay, through a receptor, a class of receptor called integrins. So, what are cellular fate processes? We will discuss in the next, like in subsequent sessions. Cellular fate processes basically refer to the basic cell functions like cell migration, cell proliferation, cell differentiation, cell death, okay, then cell spreading, cell adhesion. So these are few cellular fate processes that a cell will go through anywhere, any cell type and any tissue. Without these processes, you cannot understand whether cell is living or not. If the cell is living, they will go through these processes. So how these processes will take place, that control is with the ECM. So ECM is the components of the ECM is modulating the strength of the substrate, right, on which the cell is adhered. So it is kind of modulating the cell spreading phenomena, okay, or cell adhesion phenomena. In many literatures, people have reported by modulating the composition of the ECM, and that will indirectly change the strength of the material. The cell differentiation, a stem cell is differentiating uh, different kind of cell types. Okay, so if the strength very matlab strong or mechanically hard surface is there, then they have seen that bone marrow stem cells, they are differentiating into osteoblast, right? If the substrate is relatively very softer side, it will differentiate into adipocytes. So these kind of control ECM do have. Okay, and the control is through a particular type of receptor called integrins, okay, that are present on the membrane, cell membrane. Okay, so in this figure, you can see how cells are interacting with the ECM components. So these are two cells, okay, and here these are the integrins. So integrins are basically transmembrane protein, where some part of the protein is inside the cell and some part is outside, like outside the cell membrane. So these are called transmembrane proteins. Okay, so through this ECM, so sorry, through these integrins, you can see that this cell is interacting with the collagen fiber. All right, collagen fiber is present in the ECM. This ECM, so this integrin is interacting with the fibronectin fibers. Then there is proteoglycans. So this kind of interaction is happening with the ECM via the receptors called integrins. And there is also direct cell-cell communication. So this again takes place through a special kind of receptors called cadherins. All right, and these are the special receptors that can detect the presence of some kind of growth factors. So it is then doing cell signaling, right? This is also one kind of an ECM component. So it's a very complex thing, uh, if you so this entire picture, you can say this is the cellular microenvironment, and various complex interactions are happening at the same time within this small scale, right? So what are the important characteristics of the ECM components that we see? Whatever components that we saw, the collagen, fibronectin, uh, etc., all these are of very high molecular weight, means these are very large molecules. So if they are very large, their diffusivities means they cannot diffuse that much fast. Their diffusivity is very low. Okay, uh, means each collagen fiber, they cannot migrate, or they cannot uh, migrate from one place to another at very high rate. Whereas if you look at small molecules like growth factors, cytokines, or chemokines, they can diffuse at a relatively very higher rate. Okay, and these components are multimodal. Multimodal means ECM component. So here is the figure. So this is a figure of a fibronectin molecule. Okay, and in this molecule, you can see that there is special collagen binding site, there is special cell binding site, then heparin binding site. So there are multiple binding sites within the same molecule. That is called multimodal. Okay, so in this fibronectin molecule, uh, through the integrin receptors of the cells, they will interact here where there is the cell binding site. So this cell binding site is for the integrin molecules that are present in the cell, whereas this collagen binding sites will go ahead and interlock with the surrounding collagen fibers and make a matrix kind of a thing. So these are multimodal molecules. As I said, they can regulate the cellular fate processes like differentiation, apoptosis, migration, etc. And you can or these ECM components, they can undergo conformational change under mechanical load. So if that happens, the binding sites which are there, like cell binding site or collagen binding sites, etc., they can get exposed. Many of the critical binding domains, they have been identified, okay, and being mimicked by small peptides. So what people are doing is, rather than using the entire collagen molecule, they are just using the protein sequence corresponding to the cell binding side of the collagen. Okay, and using those small peptides as a and yeah, attaching with the scaffold, tissue engineering scaffold. Okay, and then they are modulating the cell adhesion. So for example, there is a small peptide called RGD peptide, which is composed of only 3 amino acids: arginine, glycine, and glutamic acid. These three peptides. So these 3 amino acids make this small peptide. So this RGD peptide, if it is there in your scaffold, biomaterial scaffold, then it has been shown that the cells can firmly adhere to the substrate. Okay, and then once adhesion happen, the cell will spread, and then proliferation and differentiation, etc. So rather than using the entire collagen, they are using the only the protein sequence corresponding to the cell binding site. So and also, these molecules often remain bound to isolated mammalian cells, because of which a small population of integrin receptors of the cells are occupied by these ECM components. So when you are isolating primary cells from a tissue, okay, so each of the cells, and within each of the cells, few of the integrin receptors will be occupied with the ECM. Okay, ECM components like a small part of protein, collagen protein, or fibronectin, etc. So this happens, and people have optimized protocols to get rid of those or digest those kind of proteins. Okay, so these are the important features that it is important for you guys to understand in a tissue engineering perspective. So this is the image that I showed you. So till now I have prepared for today's session. If you guys have any queries, let me know. It is almost time, I think. Any queries from your side? Is anyone there in the meeting?
Yes, ma'am.
Yes, ma'am.
Yeah. So do you guys have any queries that you would like me to address?
No.
No. So I think it was easy, right, till now. So we'll continue the discussion on yeah. Yeah, Bhupendra, you have a query. Go ahead. You can unmute yourself and ask.
Ma'am, why do we need to add special domains?
So
As you said, RGD domains.
right. So if you categorize cells into two types, so there are one cell type that adheres, so there is called adherent cells, and there is one cell type that do not adhere, non-adherent. So in our body, except blood cells, all other cells wants a substrate to which they want to adhere. Okay. So for adhesion, as I told, you need the integrin receptors. Okay, these integrin receptors, they can recognize the cell binding sites in your substrate. Okay, and anchor your cells well. So if cell do not adhere, they will not going to spread. They will not gain their normal morphology of that you can see if you are looking under the microscope. And after cell spreading, you will able to see subsequent cellular fate processes like proliferation or differentiation or apoptosis. So for adherent cell types, you need to provide a substrate to which they can adhere. And these RGD sequences, as I said, they are the sites which these integrin receptors can recognize. Okay. Is
Okay, ma'am.
okay? Yeah. Okay. Any other queries? No. So let me just quickly take your attendance. I'll you can tell me your name so that I can mark your attendance. Okay. Any one of you go first, and then next person can say their name. I'll just mark your name.
My name is Murli, ma'am.
Okay. Okay. Murli, let me mark your name. Okay, Murli. Who is next?
Nidhi Priya.
Nidhi Priya. Okay. Next.
Kirtan.
Okay. Kirtan. पहले मैं लिख देती हूँ. Kirtan and Janvi. Okay. Next. Is anyone left?
Satyam Bhatla.
Harshita.
And Harshita, right? Okay. So thank you guys for joining. Okay. I think you guys can now leave the meeting. I'll upload. Yeah, yeah. I will
Thank
upload
you,
the
ma'am.
recording and as well as today's
Hello.
meeting notes. So tomorrow there's going to be a small quiz. The quiz, you don't have to worry about it too much. If you have understood the topic, you will be able to do it. So it will be 15 minutes quiz around, and all the questions will be like MCQ types or one word answer kind of a thing. So it will take around 15 minutes of tomorrow's session, and then we'll continue with the topic. All right. So I'll see you guys tomorrow.
Ma'am,
Yeah. Yeah. Yes, Janvi.
ma'am, Bhupender, yes, ma'am, Bhupender attended actually, his mic is not working.
Okay. Okay. Bhupendra. Okay. I'll mark his attendance. Okay. Thanks. Thank you, Janvi, for informing me. Okay, Vani, your attendance is also not marked. Okay, let me mark it. Okay. Okay. And also tomorrow's session will be from 5 to 6 p.m. Okay, like regular. So only today's session was from 7:30. Right. Thank you guys for joining. I'll see you guys tomorrow. I'll end the meeting. Okay.